Hello everyone, in this video we will talk about RNA interference technology or RNAi technology. So what is RNAi technology? RNAi interference technology is a biological process in which RNA molecules specifically select and inhibit gene expression, typically by degrading a target RNA molecule. It's a gene silencing phenomenon in which double-stranded RNA suppresses expression of a target protein by stimulating the specific degradation of the target mRNA. Actually, what happens? mRNA molecule that is produced in the nucleus, suppose this is the mRNA molecule produced from DNA in the nucleus, then it is transported to the cytoplasm as a single-stranded molecule, then it is translated into protein, right? So this is the general idea of transcription and translation of a DNA molecule. But somehow if this mRNA molecule form a double-stranded form, like if it's suppose it's both sense and antisense RNA are transcribed, they have complementary sequence, they might form a long stretch of double-stranded RNA molecule, or sometimes it may happen that the mRNA molecule have so much complementarity that they will form some loop-like structures. They will band and bind with each other, okay, and form loop-like structure, or they might form some hairpin-like structure like this because of presence of repetitive sequence, such as in case of transposons, inverted sequences, and all. They can form different double-stranded structures, okay. These are double-stranded RNA molecule. So whenever there is a double-stranded RNA molecule present in the cytoplasm, then the RNAi mechanism come into play, okay. Many organisms uses this RNAi mechanism to control genes, and it can also be used as a tool in the laboratory as a therapy, okay. So when the cell contain double-stranded RNA molecule, different enzymes will be recruited by the cell so that it can be processed into three different types of small RNA molecule, like you can say miRNA or microRNA, siRNA or small interfering RNA, and shRNA or short hairpin RNA. This type of structures are formed by processing the double-stranded RNA molecule, okay, depending on the structure of the double-stranded molecule, okay. Different types of small RNA molecules are formed using different enzymes. So what are the enzymes actually involved? I'll talk about it a little bit. Different enzymes are involved actually in processing of this double-stranded RNA molecule, such as DICER, okay. DICER is a RNase 3 kind of enzyme, RNase 3, okay. It's a RNase 3 type of enzyme which acts on a double-stranded molecule and cleave both the strands, so that small molecules of the RNA strands are produced, okay. They are fragmented, you can say. DICER molecule fragmentize a long stretch of double-stranded RNA molecule into small RNA molecules. This is the function of a DICER molecule, okay, resulting in the formation of various type of small RNA molecule. There is another enzyme involved which is called RISC. It's a multi-protein complex, okay. There are a lot of proteins involved in formation of RISC, multi-protein complex. It's not a single enzyme. There are a lot of enzymes involved, like your DICER will be there, then your Argonaute protein family will be there. Then your RNA, this double-stranded RNA that will be formed by processing with DICER, that will also bind to this RISC protein, and they together form the RISC protein family, that is RNA-induced silencing complex. This is the actually main protein, okay. This is the key player, you can say. This is the RISC protein is the key player in RNAi mediated gene silencing mechanism. This will break the double-stranded small RNA molecule into single-stranded one. Then the single-stranded molecule will be directed to the target mRNA molecule that is supposed to be cleaved. Then with the help of RNase H kind of enzyme, it will cleave the target mRNA molecule. So RISC is the key player in RNA induced silencing mechanism. I have mentioned about RNase 3 and RNase H type of enzyme, right? So RNase 3 is a endonuclease enzyme that binds to double-stranded RNA molecule and cleave both the strands. It will bind to the double-stranded molecule and cleave both the strands, resulting in formation of small double-stranded RNA molecules. And RNase H is another kind of enzyme that can bind to a double-stranded RNA molecule and it cleave only one strand, okay, and degrade that strand so that one strand remain intact, the other strand remain intact. So this is the idea of RNase 3 and RNase H kind of enzyme. RISC will have RNase H kind of enzyme that will ultimately break the target mRNA molecule when the your guide RNA molecule guide strand will bind to the target mRNA molecule. Okay, I forgot to tell you about guide strand and a passenger strand. So your miRNA will have two strands, right? miRNA or siRNA or shRNA, whatever it is. So after processing, this small RNA molecule will have two strands which will be taken up by the RISC protein, which is combination of many different protein, as I have already told. And out of those strand, one strand will be called your guide strand and the other strand will be called your passenger strand. The passenger strand will be cleaved, okay, before taking to your target mRNA molecule, and the guide strand will remain intact with the RISC protein. This guide strand actually is the antisense RNA, you can say, which has complementary sequence with the target mRNA molecule, which is only needed for target selection of the mRNA molecule. It will actually have complementary sequence with the target mRNA molecule, therefore it will bind to that specific site and it will direct the protein that is present in the RNase H protein that is present in the RISC enzyme to cleave this mRNA molecule. Okay. So this is the basic idea of RNA interference technology. That means what? Your double-stranded, if your double-stranded RNA is formed, it will be processed by various enzymes like diacers, RISC protein and all into small RNA molecule. Then these RNA molecule will be directed to the target mRNA molecule with the help of some protein complex called RISC protein complex that will process it into single-stranded mRNA molecule, and it will have complementary sequence with the target mRNA molecule that will bind to it and ultimately trigger its cleavage into small fragments, and these small fragments will be then degraded so that no protein is formed. That means what? A gene expression is completely blocked, okay, for that particular mRNA, because this mRNA is degraded, so no protein will be formed for that particular gene. This is the basic idea of your RNA interference technology. In my next video, you can see the mechanism of RNA interference technology by siRNA, miRNA and shRNA. Thank you.